many of us, I did get looped into the whole deep learning stuff, at least for more in NLP. Yeah, so like I said, I'll be going a little bit about the relationship, the theoretical links in biology and machine learning and NLP, a bit about deep learning and BERT, which I can skip over super fast if we see that it's a bit redundant. I'll work with ProteinBERT and some fun stuff with anomalies and protein language models. And if I am speaking too fast and somebody, raise a hand or ping me or whatever, it's a bit of a problem for me sometimes. I apologize in advance. Okay, so wee bit of biology 102. So proteins are, like there's a saying about how DNA is the language of life. DNA is the you could call DNA is maybe the hardware or the circuitry. Proteins are the machines. They are the software and a lot of the hardware and the machines of life. Basically, DNA. Why is DNA important? Because it tells the world, your body, whatever, which proteins to make. Proteins are molecular machines. They're very, very long strings of molecules called amino acids, which naturally fold in on themselves, around on themselves, and create very, 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 very complex and energy efficient machines, which do things. Like your cells are made out, the walls are made of proteins. Proteins bring things from point A to point B. They disassemble stuff, they build stuff, they copy DNA, they write DNA, they melt stuff, they do whatever you want. And again, at the end of the day, though, there are very, very, very long chains of, like I said, of molecules called amino acids. So, just one second, I want to see those. I'm saying okay, yeah. So, we can think about text and proteins, though, in a very similar way. In both words, we have what I like to think of as global tasks, for example. Stuff like, in text it might be, given a sentence, what is its sentiment? Or is this text a good or a bad text? And we also have local tasks, like, given a text, is this word, is it a named entity, is it what type of part of speech, is this a company name, so on and so forth. And in proteins, we also have similar types of properties we'd like to know about. At the global level, it might be, where does this protein go? Is this how stable is it? What organism is it from? And at the local level, we also have different things, like, say, for every single position in a protein, does it undergo some type of chemical modification? What type of structure does it have? Like I said, proteins take on various two-dimensional and three-dimensional structures. Does it have various post-translational modifications, basically where the proteins can undergo additional modifications, lots of different things. And really, again, they're both, basically they are sequences of characters. So let's see where some of the problems start. So in texts, the basic task we have is, tokenization. Given a text, we want to split it up into tokens. Tokens, I like to think of it as roughly the fundamental unit of information, where it's a little bit arguable. You might say it's a word, is it a character, is it a subword, maybe it's the root of a word. But in many cases in human text, we'll say it's a word, give or take. But in proteins, we don't really know what it is. Proteins are made out of, like I said, molecules called amino acids, of which you have 22. But each one, we represent it with a letter. But in some cases, a protein might function on structural units of multiple amino acids. It might be 2, it might be 3, it might be 5, it might be 7. And in other cases, there might be functional parts, like here is an area which something docks and binds to. It might be from one position, it might be from multiple ones around it. It might be because it's in a region with many amino acids, with property. We don't know what the words are or the characters are. They might overlap depending on how you're looking at it. Like, a little bit like, say, Chinese or Japanese, where you can just stick words together however you like, only it's much, much, much worse. So we can, of course, approach these with, classic approaches, everything from, bag of words and n-grams and TF-IDF, and some fun stuff I made once called mirrors and k-mers, where we don't care about local order. All of the different approaches we can do in classical machine learning. But of course, we want to do more than that. So, embeddings. So I'm not going to, given the audience, I am not going to start explaining word2vec again. I'm pretty sure everyone knows it. Actually, show of hands, is there anyone here that when I say word2vec and embeddings does not have it coming out of their nose? It's alright if it is. Again, I don't know the background of the audience necessarily. Anyone want a quick one-on-one about the concept of embeddings with words, word2vec? Okay, I'll take that. Either as a no or that I've already killed everyone. I would just note that again, when we're doing word-to-vec embeddings of, say, when proteins and amino acids, they actually, similarity between the different amino acids using word-to-vec is very similar to what we can get from looking at evolutionary similarity. You know, how similar words are in terms of how they change and replace between, say, a human, the monkey, and yeast. Then we find that the embedding similarity for amino acids from evolution is very similar to what we would get from just looking at word-to-vec style approaches. It's very neat. Also not citable because it's just some code I did once, but it's neat. So, all that said, proteins though are not a natural language. But human language is much more similar to whale song than it is to proteins or DNA or RNA or any of those, because there's lots of reasons. Basic one I would say, not only can we not speak or read or write them, there is no conscious mind which made these languages. Whales are living biological beings and they made a language. Humans have languages. Even when we make up a language, even if I'm making up Dothraki, it's still, you know, it's a language of rules. If I say I glibglau the Schnitzelfassel, you can kind of guess what I'm talking about. You have a concept of words, nouns, verbs from that. When I'm looking at a protein or DNA, was not made by anyone in order to speak. It evolved from a huge, literally huge soup. There's also the fact that, say, we really don't know what are the separators here. Yeah, you can say, well, you know, in Mandarin we don't know how to split things up into words well, but you know what the words are, even if we're not great at splitting them up. Here, nobody really knows what's a word versus a sentence, a paragraph. You know, we make analogies, but we can't really say here in this protein, those are the words, those are the sentences, those are the paragraphs or documents. They also have different statistical properties. Stuff like, say, the entropy of the sequence is different, which is neat. The length varies far more. The shortest protein is 4 amino acids. The longest is over 40,000. In natural language, you would not have, say, words which vary by 3 orders of magnitude, give or take maybe in chemistry. And even then, I don't think it's quite that bad. And finally, maybe the most important difference is, at the end of the day, language is completely made up. It's made up, semiotic, what have you. I can make up any language I want. I can make it do anything I want. Proteins are physical machines. It is a physical machine operating under constraints of physics, of mechanics, of entropy, of you name it. It is a physical machine like a crane or like a motor or an engine, like a black hole, and it operates under those rules and not under, you know, language rules. So anybody wants, there's a very nice review we did about the NLP and machine learning, protein sequences, and also feature engineering techniques for all of that. So questions about biology and proteins and all of that. The end, the beginning, the mid, the half of the intro. Questions?
One question. So basically, like enneagram approaches and stuff just doesn't work with proteins.
I'm sorry, could you repeat that? I'm just turning up the volume.
Sorry. So basically, like enneagram approaches or like grouping of proteins just doesn't, like, those are like a big no-no for proteins.
So they work really well. If anyone who did say a degree in computer science, and you remember in algorithms you have dynamic programming, recursive stuff, and there's the blast, the thing where the matrix goes backwards. The reason why it's popular and why it's so taught and where its biggest impact is actually in proteins. We're doing alignment between sequences. Local alignment actually works very well. It's not n-grams, it's doing over it's a bunch of sequences. Local similarity works very, very well for a lot of problems, not all problems. Like if I show you 2 proteins and they have the most similar sequences, most cases they are similar, but depends on what. In some cases, if they are more than 50% similar, they'll have the same function. Other cases, here are 2 which are 99.9% similar. They're different by 2 amino acids. Also why, well, why your baby is going to die, because that mutation means that it can't grasp cysteine at the end, and then basically horrible things happen. The problem is that similarity can really vary. Sometimes it's by structure, by function, by sequence, it varies. Like local n-grams work very well for a lot of problems. Like RORs, they work great, so does BLAST, but which problems and how problems and what you're trying to do. Like in general, always n-grams is pretty good, has to be said. But it's not quite enough.
Got it. So basically, like, the motivation to go deeper is mainly for like finding those small differences, kind of.
It's a mix of the small differences, and there were many problems where the similarity is not the same. like there were some problems where, again, if I'm two sequences and they're like 50% similar, 70%, I can say, yeah, they're both doing the same. But there are other problems, like say neurotoxins and neuropeptides, stuff which affect the brain's behavior, where I can say, well, it can have between 1 and 20 copies of a gene. Gene can be completely different. The order can be different. The amount can be different. They can also have completely different sequences as long as they have a same function of being short and wobbly. Because the function is to let a bunch of short and wobbly things in to just fill up an ion channel receptor. And it can be like saying, well, it can be of any type of picture you want in some cases, as long as there's lots of at least 74 green pixels somewhere in the picture. So, for some problems, a CNN will work great. And then other ones, like CNN will be absolutely terrible for that because it's doing the average or the max of the pool and it's missing out on that. But just counting the pixels would work better. So in other words, it really, really, really depends on the problem and on the context. Like if you're trying to do something really simple, start with BLAST, just counting the aligned sequences, all of that, it will work very well. And in other cases, biology is nothing but gotchas. It's like it's the elephants all the way down in terms of like maybe sometimes this is better, sometimes that is better. But the deep stuff is interesting because it's a lot more efficient, and in many cases, it can do stuff that works much better that we couldn't do alignment. It depends on the problem.
Got it. Thank you.
Any other questions? Point?
Is protein always one-dimensional, like text? Does it, or could it also be,
So technically, yes. That's a little bit of a cheat because after the protein is translated from DNA and undergo modifications, which can be additional modifications on the position. Think of it like, say, here is a word and I cover the word bright red. So in text, yes, it's still one-dimensional text. But if I give you a text to say, here's the bit where it's colored in bright red, then that's not exactly one-dimensional. But you could say, well, I could represent it in another way, blah, blah, blah. But, if I gave it a ChatGPT, count with the words which were painted bright red, it's not going to be able to do that from a one-dimensional representation. But in general, it is created as a one-dimensional sequence, which then just folds in on itself. So, there's a nice case study about origamis, which I recommend on this topic.
I see.
So basically, it's one very long sequence which folds in on itself to create 2D and 3D structures.
Okay, gotcha, thanks. I wondered if you're going to talk about like actual tokenization that you used, but is it similar to like sentence piece or word piece or byte pair?
Yeah, I'll get into that in the later bit.
Okay, thanks.
I think I'll continue. So language models.
Sorry, there was one question in the chat also about that I'm interested in as well, which is that in the encoding that you end up doing, is context relevant, or is that one of the reasons why you were saying that?
Yeah, we do context dependent embeddings. I'll talk about that in a bit.
Okay, thank you.
No worries. And also, I really recommend write the questions in the chat. That will it'll be way easier for me just to scan it from the point. So language models, I am going to skip over this super fast because I'm guessing also that most people know it. Just it's always worth giving the context a tiny bit. Again, in language models, we want to get the most likely word. We can't get the perfect answer, but cat sat in the mat is more likely than Cthulhu sat on the mat. I'm not sure if cat or dog is better without more information, but Cthulhu is still possible, and running should not appear because it's not grammatically correct. And again, state of the art in most cases is bidirectional modeling or math text, where I just hide some words in the middle and fill in the middle approaches. And this is also, by the way, still tends to beat the crap out of the next word prediction when you're not doing generative tasks. Like ProteinBERT still beats GPT-3, though ChatGPT-4, I haven't seen that compared. So, and of course, like many of us probably know, why do we care about this? Because knowing the most likely word in the middle of something means learning a lot about how the language works. Saying cat sat in the mat means I understand that cat is how nouns work and verbs work, and that cat is an animal that can sit on a mat. The relative size of cats and mats, that sitting is something a cat can do, that cats are domesticated animals, usually in houses. Like from this I can tell this is probably inside the house or a huge amount of stuff that it needs to learn implicitly in order to do this. Can someone just turn their mic off? Can't see where it's coming from. So this can be done in and why do we like this? Because this can be done in an unsupervised approach, aka I don't need to start paying a bunch of expensive people to label my data. Instead, here's a bunch of tokens, I'm going to mask them out, learn on them, and then here's a small label data set and learn on that. The supervised way. It's more fun than, say, what I'm currently doing with medical stuff, where it's very, very, very expensive to have anything labeled, and there are no free tokens. About self-supervised learning, because we want to do transfer learning, aka train on some huge lump of data, then use that as a model, use that as a feature extractor on additional tasks. And we really like this because this makes it somewhat less of an art and less of a science and more of engineering. Like the ImageNet moment, what really made the big jump in deep learning, I would argue it's arguably a tie or by between, hey, this CNN stuff really works well on this problem, and hey, we pre-trained it on ImageNet, now we get better results on just about everything. AKA training it in advance on a large task and then getting better results on any task you choose, pretty much, in a relatively predictable fashion. That's in vision, also in text. Yay, Jeremy Howard, he does not get anywhere near enough credit, I will note. Well, again, basically pre-training means we get better results from less data. At the same data budget, we see better results, so it's a smaller difference when there's a lot of data, but it's a much better difference when there's very little data. Or in other words, it works, it makes it learn faster, it makes it learn better, it makes it learn better from the same amount of data, and it's even more so when we have a small amount of data, which again, in biology or medicine, you have like, the chart here, this is from a bunch of NLP tasks, like in biology it might be single digit to dozens of examples. So the chart here does not go left enough to show the differences in the amounts we're talking about. Again, as we all know, bigger models, better models, more pre-training, better results, yada yada yada. And very popular with Facebook, they did pre-training on everything from Instagram with billions of images and showed an extra X% improvement. Never mind all the current stuff with the huge language modeling with which I'm sure they do not cheat on any trade sets or evaluation benchmarks in any way. But yeah, but basically there's a huge trend towards very, very, very big, very expensive models. I will also note I am from academia, specifically a small computation biology lab. When I say small, I mean we had one GPU which we bought halfway through, and most of the time I was using my stuff at home, which when I was not using it to play Doom during Corona. Excellent game, but still. So now, any questions about the whole yay pre-training paradigm is great? Thought as much. Okay, so we're getting onto the actual bits of the meat and potatoes. So ProteinBERT. So the idea here is, let's do a self-supervised model for proteins. And so, like this, I started working on this in early 2019. Came out around 2020, 2021. Corona made it a bit delayed, as along with some family members dying. So at any rate, in protein language models, the general idea is, let's do the same stuff as in NLP. Take a language model, do self-supervised pre-training over lots and lots of data, and then use it for some other task. Yay, profit, better results. And when I say recipe, I mean carbon copying of recipes or models, say from Hugging Face and the like. I'll also note when we started working on this, the Hugging Face did not play nicely with protein data sets or training from scratch or doing pre-training from scratch or adding additional tasks. So we ended up doing all of this in Keras. It's supported. But basically, most of the also earlier works were just really a copy-paste or do the exact same thing as NLP. Many cases also take the biggest data set they could find. And afterwards, there are some different benchmarks, which are very nice for evaluating fine-tuning. So somebody ask a question? It's not. So, there are lots of different models coming out. I'll note, among others, say Ross Lab, Protrans, ESM2 from Facebook, though they shut the team down, though I hear they're raising a bunch of money for their startup. And lots of other works which have since come out. If I were to do this slide now, I'd need another couple of pages. So ProteinBERT, what did we do, which is interesting? So one is that we have two different very, very novel pre-training tasks, which I'll go into in a bit. One which is around the global task, and another sequence level task, which is a bit inspired by a lecture and replace token detection for those who know it. Additionally, our architecture has very, very little to do actually with BERT. I use the name BERT because I said it's going to be way easier to explain and to sell it to people. No regrets in that regard. The only thing it has to do with BERT pretty much is that roughly maybe masking in the middle and technically attention, sort of attention. So what does our architecture do? One, we use a global attention with linear complexity instead of n squared self-attention. Our model is super tiny. It's 16 million parameters, 6 layers. It's supposed to say 650 million for a BERT equivalent model there in the field. Oh, sorry, I thought somebody asked something again. We use a lot of convolutions and try the dilated convolutions. We are flexible to sequence length. We don't use positional embeddings. And we support global and sequence level inputs and outputs. And finally, we show very strong results on a bunch of different benchmarks, despite being much, much, much smaller in everything you can name in terms of flops, pre-compute time, evaluation time, and so on, compared to anything comparable. So how one of the layers, we have 6 layers. So the model takes in inputs at the sequence level, i.e. it's a sequence, and also the global level, which can be a bunch of annotations, or they can just be all masked out. Does compute, and outputs representations per position, i.e. at the sequence level, and for the entire sequence, i.e. the global level. And I mentioned earlier that we use a linear attention. So what we did, we kind of came up with it independently at the time, which is a bit of a bummer because I found out that we'd kind of come up with it independently because I've probably gotten a better paper out of that, where basically we have a global attention layer. So classic self-attention n squared is each position use attention to all other positions in a sequence, which means if I have twice as many positions, I need to do 4 times as many computations, which gets expensive fast. I want to be able to work on a sequence with 2,000 positions or 4,000, since a protein which I chop it in half is, sometimes it can be a bit of a problem to work out what's going on. It's not like a Wikipedia article where you just to get the title and the header, and that's pretty much enough information in many cases. So what we have the global attention basically is we have a layer which is attached to, so I'm not going to go into the exact details, but basically we have a single fixed size fully connected layer connected to the input and annotations from the global and the local level, and it pays attention. Each position in the sequence is compared to that. So we're doing attention versus that single sequence and not versus all other sequences. This means that if I have 10 times as many tokens, I'm only doing 10 times as many computations instead of 100 times as many. Big difference. Also means that this global attention layer is very good at getting input from everything at once. We found it's very efficient. And the best part is that it's very, very interpretable. So I was trying to look at which position, at self-attention versus which position. I'm looking at the attention over all positions and just see how that changes. Again, this is also, by the way, multi-head attention, so you can look at how many attentional heads for the global attention, never mind for the deeper layers. There's more about it in the paper. I'm not going to go super, though I will show an example. But also note, we don't use positional embeddings. We found that the convolutions, we use a bunch of strided and dilated convolutions, actually work better. We don't need the positional embeddings. This means that if you want to run it with a sequence of 20,000, then it'll work, and you don't need to save, an embedding of length 20 or 40,000. And again, like this is just really fast and small. Convolutions are efficient. So the sequence task is it's kind of like over time I found later it's kind of like T5, a little bit like Electro RTD. And the idea is instead of replacing some tokens with a mask token and then predict that, we randomly replace some of the tokens with another random token. By the way, here when I say tokens, I mean amino acids. Our model looks at the token level, the amino acid level. Each token is a single amino acid character. So it's basically character level tokenization, not a sentence piece or any of the other ones. We actually looked into them at the time and we just got a bunch of bugs. And we decided that also for some task, like say predicting at a specific position, chunking a bunch of letters together would harm it. Also supported by some later work with it. Some people try to test it. Basically, you care about the letters even if it's not necessarily as efficient, but you really need it for some of the tasks. Basically, a character level tokenization, to put it that way. So again, basically we take up the sequence, about 15% of the tokens we randomly replace, and the model does not know which characters were replaced. It needs to predict for each position in the sequence what the original amino acid character was. So it's not trying to predict, oh dang, it scrambled up the text here. It's not trying to predict 0 or 1 as an RTD was just replaced. It's trying to predict what the original amino acid was, which is a much harder task. But on the other hand, here we have only 20 amino acids and not, 32,000. So it's easier in that regard, though it's a very different domain. So this is the big benefit versus MLM, rather like an Electro, where we're learning over all positions in a sequence, learning more of the everything to everything, of the embedding and relations and the context-dependent embedding, as opposed to only learning on the 10 or 15% which are masked. Questions about the lecture, RTD, take 5, on this task so far?
One question about in the previous slide you mentioned Go input annotations, right? So
the input annotations I'll go into in a sec.
Okay,
Next
good.
slide. Here it's a vector sum, it's binary cross entropy, by the way, and also the second task which I had.
One question. Sorry. One question about this task. So is it possible that you randomly replace the tokens and you get an entirely new valid protein, or it's possible for a single?
It's completely, replacing a token does not mean that it'll be invalid. Actually I did a bunch of work in the past about how there's reduce amino acids to similar ones because they have different physicochemical properties. In many cases you can replace it with a similar one and nothing will happen. If I replace K with R and R with K, in most cases it won't matter. In some cases it might slightly reduce binding affinity, but it will be instead of 87% it's 84%, which is binding probability in X moles of something, which doesn't really necessarily matter that much. But if I replaced a glycine with a proline, it's I basically, it would be like if I replace your eyeball with your thumb. It's not supposed to be like that.
Okay, makes sense.
That's a terrible example, but it's very accurate. It's some of the places might be very bad, and again, the general idea remains, just add in noise. Let's add in noise, and it needs to noise it. And again, we actually had some ideas about how to do this better, but there was a huge, just adding in a novel pre-training task was just a huge amount of work. Also, when we did this, we just didn't have the infrastructure. We did everything from scratch in the KRS and just so much work. The raw data is very, very noisy. If you were doing today, it would be a fraction of the work because it's been a lot more stuff done. But back then we were we were almost in the first teams to do a protein language model. We got scooped by Facebook and Google and another team where they had, well, I'll get into this later. But basically, doing your own pre-training task is very interesting. It lets you do really cool stuff. It is a huge amount of work, and I understand why people don't do it that much. It's it's a huge headache. Okay, so the global level task is, so in biology we have something called gene ontology or Go annotations. So there are basically many thousands of manually curated annotation labels, cellular, biological, and molecular function. Everything from where is this protein in a cell to this is a protein which is involved in snipping on a dibasic cleavage site to this is involved in cleaving hormones to this is involved in the citric acid cycle. Yay lemons. Burn it with the lemons. So there are thousands of those. They're arranged in a great big DAG graph for anyone who likes network graphs. And the problem is though that they are annotated on proteins in which they were found. They are not annotated if they were not found, and there is very partial annotation because this is from biology. It is expensive, it is hard, it's coming out of labs. It's not. Some of them are easy to check, but still it's out of a lot of proteins. Also, they are very noisy annotations. So you have out of all of our data, only 43 million proteins had any type of Go annotation. Out of those 43 million, they had about 2 annotations on average. Keeping in mind, out of these 8,943 are the common Go annotations, which had at least a minimum of, I want to say, 100 occurrences in the data. There we did it with as a multi-label task. And this is multi-label with unknown negatives and incredibly noisy labels. So really, really noisy, really, really sparse. Zeros are ones, zeros are not necessarily zeros, and most of the labels are going to be 99.99, I think an extra 9% zero, and very, very, very noisy task. But they are pretty much the biggest lump of annotated data we have over proteins, more than manual annotations or whatnot.
I had a quick question about the tokenization. So, in a lot of these proteins, I assume most of the actual sequence is just forming like basic secondary structures. Did you ever use longer tokens than single amino acids, or you only use single amino acids as tokens?
Only single amino acids in this version of the problem. We wanted to do to experiment with like multi-sentence piece or whatever, but we just didn't have the budget, mildly.
Okay.
Actually, it's a work I saw recently by some colleagues in Tel Aviv where they tried the larger vocabulary and language models. They actually got worse results, but it might kind of depend on your task. It would be more efficient, and there are some, just running it yourself, you will see some very efficient compression. But the problem is for some tasks you will lose a lot, because if I were to tell you to, do sentence piece, but we can't really tell words apart, and it is a company name, and you are squishing the company name in something which is not a company name, that's going to mess up your results. Have that as a task, as well as, I don't know, how many words are in this text. So for some tasks it will work better, for some it will be much, much worse.
Okay.
And so far there's no free lunch in that regard. It might be a better trade-off, but yeah. Oh, and for anyone else, we don't use an encoder-decoder approach. Our generator was just randomly sample an amino acid and replaced one. Like we wanted actually to check. It's not even. I'm not even sure if we did weighted in the end. Very simple. It's very stupid, but it works quite well.
Do you know if anyone has incorporated like MD with this to try and like change the tokens based on like what structures actually form or something like that, or not really?
That's a good question. So as far as I know, no, nobody has done the MD stuff. There has the diffusion, people doing work with diffusion and structure. There's a really nice work on mapping from sequence to structure, then from given a structure, what's the sequence. But pretty much just about all of the other work I've seen done in the field with the pre-training tasks, either they copy-paste of BERT's MLM or next token generation because they want to get money out of people with models. Much. Like I haven't seen anyone using our approach or even a lecture, honestly, mainly because it means a lot more work. Like even say, I was that random example, I wanted to try the BERT approach, but they don't have the pre-training code out for doing that with the RTD task, and just it's a huge kludge to implement it properly at scale. So when I say at scale, like the amount of data we had. So for our pre-training, we used 1/100 million proteins. Each protein again is on average 300 to 500 amino acids long. Roughly, most of the shorter ones were around 100 and something. The longer ones could be up to tens of thousands long. It comes up to a few billion tokens. And again, I'll note that this is actually out of a data set called UNIREF 90. So in proteins, I mentioned we have lots of proteins, but we have hundreds of millions of known protein sequence with different levels of confidence. It might be, did we see the protein? Did we sequence it? Did we have the DNA? Did I find a copy in DNA, which I find in a lump of mud which looks like it? And so there are lots of different ways of saying it. Like everything, like in text, we have people taking C4, C3, and some people who take all of it, some who clean it more or less. And I will note the people doing better these days are the ones who learn from bioinformatics, because in bioinformatics we say, well, lots of these are redundant. I might have, say, thousands of proteins which are just HIV plus minus one amino acid. And it's, Sorry? A question?
I think that person accidentally had their mic on.
Okay, so basically we have some proteins which are very, very, very similar. Like I said, proteins are evolved, and we might have the same protein in lots of different species. We might have almost the same protein in many species. We might have protein with important differences in different species. Like we have a bunch of ones which are basically the same as in snake venom, which are used in our brains for lots of neat stuff. So there it's undifferentiation. So what we have in proteins is something called the UNIREF. It's basically clustering sequences by similarity redundancy. UNIREF 100, 90, 50, also some other ones where keep a sequence out of a cluster of sequences, keep one representation per cluster, which has at most X% similarity to the others. If I weren't using any clustering, I would have at about 250 million, and with 90% similarity, we're down to 100 million. 50% would be, I want to say, 70, 80 million, 50, 70 million, something like that. So it's, again, this is really, really reduced to redundancy. We could have gone a little bit lower, There's a bunch of biological reasoning behind this, by the way, in terms of function versus structure and how that's maintained. I'm not going to go into it. And like I said, out of these 100 millions, only 40 had any annotation. Out of those, only it's an average of 2. And then annotation, by the way, can be stuff like, this is from a human, this is from a virus. So, you know, or this is from something which has been studied. So most of those are like really terrible annotations and lacking annotations. In terms of compute time, we trained it for about 3.5 weeks on one GPU, a 16 gig GPU, none of your 80 gig fancy nonsense. This was enough time to go over everything 6.5 times, 670 million. This is much, and on basically on a home computer. This is just stupidly faster than comparable works. Just say Facebook's ESM2, by the way, excellent model. They used, if I recall correctly, 500 GPUs for a week or so on a nice optimized setup. So to go over about a third of the amount of data. So again, it's about 30-70 times slower. And if you just done this on an 80 gig GPU, it would have been a lot nicer. So yeah, this is much faster, which makes it way more attractive for anyone trying to do their own experiments in biology labs. Like, I had a conversation with a student of mine a week ago about how, you know, why you're having trouble with this table and is it so big? And then she said, yeah, no, but I have a good computer with it. It's got 8 gigs of RAM. And I was like, what year is this? Yeah. When I said it's small and fast, I kind of mean. In terms of the training tasks, so we actually ran it multiple times with different sequence lengths. They basically, you know, context size given, 128 in blue, and also long ones, 512, 1024. You know, there's pretty much no difference between them. In the longer ones, again, average sequence length is 300 to 500 or so. So when you have most of this time you have the entire sequence in, then that works pretty nicely. When it's a lot shorter, then it's like, yeah, big difference whether you can see the whole sequence or not. In terms of the sequence task, you can see that it just doesn't saturate, keeps getting better and better and so on. Again, this is over 600 million, so it's over about 6 epochs. After going all over the data 6 different times. So, you know, 1 epoch is not necessarily enough. The annotation loss does kind of saturate around 3 or 4 epochs, which is still, you know, it's going over all of the data 3 or 4 times. Your average pre-training setup is we went over X% of the data until we ran out of money. And then there, it's kind of the annotation. It definitely does we see a lot of saturation, also noise. My guess about why it saturates more, by the way, is just that it's from manual annotation. We have much less coverage there. And just, you know, at the end of the day, it's, again, 99.99 something percent are all marked as zeros. They're not really zeros, but we don't know, and the model can't know. And, you know, just that it's hard to do that much better than saying, here's, you know, 8,928 zeros in a row and some ones, and to do that much better than that. It does do better, and we go into how much it helps, but it's, you know, tricky. And okay, any questions about the architecture before I go a bit more into the results? Okay, so I'll go into the results and then we can go over a lot more of the stuff a bit more in detail before the last bit. So again, these are some benchmarks called TAPE. They go over a bunch of different tasks. Lovely paper, lovely evaluation. Also extremely annoying that a bunch of the other works which came out a bit later decided to, oh, here are the benchmarks. We're going to, instead of using accuracy, well, let's use a different metric because, you know, we just don't want to be compared on the exact same numbers. Which, you know, is a very bad code smell when you see someone saying, oh, no, we just want to use a different metric to evaluate than in the benchmark. It's like, you know, especially when you can't actually run that stuff yourself properly because it's... So anyway, here we have a bunch of different models compared with and without running pre-training. So fine-tune it on the task with the pre-training and without the pre-training, top and bottom. TAPE is a classic transformer, pre-trained on a bunch of data with a lot more compute time than us, by the way. Like full classical transformer, an LSTM, which does, by the way, really well, and our model, and 2 more bonus ones at the bottom. T5 is a T5 model trained on a supercomputer with they have a few models with data point being a budget of, I can't remember how much, hundreds of thousands of dollars, if not more. That's the one I mentioned from Google. There's also, I didn't put an ESM here, but yeah. So the tasks we have are structure at the local level. Secondary structure is basically for each position in a protein, what secondary structure, what 2D structure is it in? Is it like this? like this, it's like that. Okay, I can't bend my fingers enough, but basically, think of it a bit like, say, doing parts of speech tagging or the like, only more important. We have remote homology, which is multi-class few-shot learning. We have in proteins, we have, you could call them families of folds, which is saying, like, this is an elephant, this is a cat, this is a dog, this is a flea, and we have cases within them, but we have a couple of examples. Here is a new animal. Is it more like is it an elephant? We have about 1,100 classes. Most of them have on average, they're about 5, 7, 9 examples each. So it's very few cases per class. And it's based on similarity at the structural level, but we're using the 1D stuff. And 2 more tasks, which are about mutation, where we take a protein and we mutate it. We have some examples in the training data, then we have a bunch of weird mutations, which is in the test data. So it's an out-of-domain task. Here's a bunch of sentences in English. Now, here are some sentences in Hebrew where we also added spelling mistakes. How is this sentence positive or negative? Very out of domain, and it's very, very hard to do. They do me, like, everything goes down way worse on that. And these are the 2 engineering tasks, like how bright does something glow and how stable is something in a bunch of heat. Bunch of mutations when you only trained on normal data. So our model does quite nicely. We don't read the state of the art in some of the tasks. Without pre-training, we actually do the best by a long shot. Other pre-training is a good proxy for just basically inductive bias. Like if I were to start on a completely weird and different task, which happens a lot more than you would think, how well is it likely to do? Like, LSTMs tend to be good for text. CNNs, great for images. Yeah, we can say here is a better example when we're using a pre-trained VIT model, yada, yada, yada. But like if I'm saying, well, okay, I have a new example from, medical imaging and 2D or 3D, I would say, well, yeah, let's start with a CNN and not with an LSTM or whatever. So we actually do get the state of the art on each one of the tasks, and in everything else we get competitive results. Like say we do better than the normal transformer on many of the tasks. We do slightly less, plus minus from the LSTM. The ProteinBERT does better, but actually didn't do on the other stuff. But again, that's a 3 billion model trained with a huge budget, and it's not one you can run on a normal GPU. Ours is, well, half the size of the normal transformer. Never mind the computational complexity cost and so on. So we like it, in addition to the other benefits like flexibility. And okay, so cool stuff about the pre-training. So, and then I guess up a bit for questions. One thing we found is that there's always a talk about how more pre-training does better, and how it aligns with downstream tasks is the assumption. But what we found was that for some tasks, more pre-training does better. For the structural tasks and the protein family tasks, they do better. Here we see basically snapshots from as we did on pre-training, and then did fine-tuning for each snapshot. And some of the tasks we see they just keeps getting better, the top 2. But for the bottom ones, one the mutation, actually some pre-training does help a lot. Like let's say pre-train on, I don't know, 1 million sequences or whatever, which is, peanuts. But beyond that, it just kind of bounces around. Look at the stability. It's just after we trained on, the 0.1% out of 100 of that amount is just kind of bouncing around there. So it really doesn't benefit much from anything beyond the very, very low level features. So that's kind of interesting because pre-training is very expensive and it's very slow, and it's saying basically, well, if it doesn't help, then maybe you don't need to do the pre-training always or to invest in it as opposed to looking at the architecture, again, depending on the task and problem. And again, we're talking about, 70%, 70, 60 correlation. We're not talking about, an AUC of 50. So it's interesting. So any questions at this point before I go into a few more of the nitty gritty?
Hi, one question. So in the architecture, the one kind of like main interesting part to me was like you added like the Go annotation. I forgot what you call it. But how kind of like I was curious, like did you test like just classifying for these benchmarks just from the Go annotations? Because I'm curious how much that contributes. Yeah.
So the Go annotations are actually, we denoise them when the model supports using them as input. We denoise them massively, like 95% annotation noising. And for the benchmarks, we don't use any of them as input.
Oh.
Because they're not used as input because that would be, we thought it would make it too easy because it can overlap. By the way, we also, all of our benchmarks, we removed anything which was similar, more than 30% similar in sequence to anything in any of the test sets of the benchmarks. Like a few, I think it was came out to a few million sequences. Again, we don't see other works doing, which to my mind is also a little bit of cheating, because pre-training is pre-training. If your test data appears in your pre-training, it is easier. You know, if you have a next token prediction and you have your tested data in there, it makes it easier for it. But we don't have, so you know, because otherwise, again, the Go annotation task, they can have labels like, you know, this is a protein for I don't know, X and Y, and that can correlate really strongly with a label of this is protein family X and that is protein family Y. So it would be cheating if we left it in, and we didn't cheat and do not cheat.
Interesting. So
I mean, cheaters we can really.
And so for like the input to the model, for the input, kind of like the model accepts like one like Go annotation for like as input, but for that you kind of just put in like a 0 vector or something during benchmark.
It can take as an input up to the vector of 8,943 annotations of 0 or 1.
Okay, and during
Or you can just
benchmark.
put them in as all zeros because it's used to them
Okay.
being 0 most of the time.
Okay, gotcha.
It's a bit.
Interesting. Okay, thanks.
Actually, we ran it at a different point, and like not giving it the Go annotations as the input actually worked better. And I kind of regret that we didn't just retrain it as that, but that would meant messing around a bit with the code and the inputs, and it's a schlep. Okay, I also mentioned the global attention. So again, this is actually quite neat. So it's, to my mind, it's relatively interpretable. So what we have here is we have on the left we have the attention which the model gives for different positions in a protein in a sequence without doing any fine tuning. We can see how it's paying a lot of attention to that D there in the middle and so on. And again, these are the different attention heads across the different layers. So again, 6 layers and 4 heads, global attention. On the right, you see how the intention works and how it's different. And this is the difference when we do fine-tuning of the overall model on a task of detecting if a protein has a signal peptide. That's a type of, basically, is this a protein which is going to be sent outside of the cell? And it's a marker. It's kind of like saying, is this a text which has at the beginning as a label of, you know, take me outside and shoot me, go on, I want to go out into the world. And we can see how the attention really changes in terms of what it's looking at, which to my mind it's very neat because you can look at it between the same head, between layers, or the same layers, different heads, different layers, same head, for example, and how it goes deeper. You see different things paid attention to, what it's looking at overall. This is something that to show it properly you need to start doing more average and sum and max between per layer or per head or whatnot. I would say that again, to my mind, this is still like a lot more readable than say everything compared to everything, because you're looking at all of the heads purpose at each specific position and not each position times all of the other positions, because it's like, you know, RAS. It's a lot harder to start doing that compared to everything, as opposed to the dog bit the policeman or the policeman bit the dog and saying, oh, well, bit and policeman to dog, but then I can see the relation. You know, which A do I look at, which R, which P, which Y, which K. So, you know, to my mind, it's a really neat interpretability approach, which, you know, really hope others would use. So I also mentioned, so we did the whole Go annotation pre-training task, which for those of you who've been paying attention, it's, you know, incredibly noisy, incredibly sparse. We basically, most of the time, we just give it a bunch of zeros. Most of the time, again, for 60% of the data, it's nothing but zeros, and it's telling it still to output the zeros. Stupidly noisy. So did this actually help? Which is what one of the reviewers asked. You know, good old reviewer number 2. And so what we did was we ran the entire pre-training pipeline from scratch twice, with and without the Go annotations. Which, if we were using something more BERT or the like, that would have meant, you know, the question? So basically what we did is we ran the entire pre-training pipeline with and without the Go annotation task. And what we found was that, yep, it does work. It doesn't help for everything. We actually ran, by the way, a bunch more benchmarks than the ones in the paper. So they're in the paper, but a bunch more additional novel ones, including some ones for my previous work. peptide one is what I did my bachelor's, master's, a lot of work on it. Like, it's kind of nice to see well we ran out of y-axis in terms of how high the score was, which is neat, very very neat. But basically the Go task did help, despite it being incredibly noisy and sparse and all of those caveats. It either helped or it didn't hurt. Like again, the red is for different snapshots, like how well does it do versus the one without. So it's either better or it's bouncing around kind of the same. So, it helps. Is it worth the amount of extra effort? That's a different question, but we're in research here. We're not asking silly questions like that, or say, should we spend the money on just annotating a few examples instead of doing a pre-trained foundation model? You don't ask those type of questions. And by the way, there's still some more stuff after this, is that pre-training does work, and proteins are similar enough to language that we can benefit from all of the hard work done by those nice people doing different architectures. But proteins aren't the same as language, and we need to know this and to take advantage of it for people who are in the field and domain. Same applies to medicine, by the way, and the like. And these unsupervised representations which we get, whether it's looking at the attention or embeddings, have a huge amount of information, and proteins are interesting and they are hard to understand. There's a lot of info there, and anything which can help us get to it is good. And I also like that, again, we used a tiny amount of resources. This is like, again, me and two friends, and then in a cave with a box of scraps, pretty much. This is not we are not a computer vision NLP lab with a pipeline set up and the infrastructure set up, or even these days it's a lot easier in terms of point the tokenizer at the data set and then run MLM on it. We did everything here, everything from scratch, loading the data, training everything from scratch in Keras back in the day. These days it's way easier. It's a lot of data. Just doing the chunking and resuming and everything and that over this much stuff is. Oof. Also, this stuff is great for small data tasks, which is what most of science and biology and research tends to be. And by the way, everything was being ported to PyTorch and to Flax, I saw. PyTorch by some nice people in Eleuther. I recently uploaded it to Hugging Face, since the Hebrew University had to close their FTPs because they, lots of unpleasantness these days. And but yeah, so you can try, and lots of people have been using the model. It's, I take it and use it. So the next step is some stuff about anomalies detection and some fun research stuff, but let's have a little bit of time for questions about the ProteinBERT.
Just to kind of like, I read the paper, and just to kind of like get my mind around the global attention, is it basically like the query matrix just kind of like comes from the global annotation and it doesn't have the sequence length? So instead of getting the ON squared like matrix in like query key, instead you get just the ON matrix, and then you kind of like do like a dot product between that kind of.
So it's doing a dot product. It's also has the local, the additional stuff put into it. Otherwise, it'd be kind of like again, most of the cases, it's not getting any global input at all. It's like densing and broadcast and just squishing it all together from the local representations before it applies them to each position.
Okay.
It's easy. Honestly, the code is better at explaining it than I am. My co-work on it, Nadav, he was the one who really came up with when we were discussing and brainstorming stuff, and he did the implementation. But basically, it's there. You can see the little arrow from the local representations are also looped in. It's not the global. When I say global, it's not referring to the global inputs and outputs. It's referring global as into the whole sequence. I didn't realize, but yeah, it's kind of confusing in that regard. Basically, like if I were to say a simplified version would be, well, let's take the embedding from all of the positions and also the global input. Then let's do a MLP over it and mean pooling and whatever, and that's the global thing. And then we do that per each position. And we go to the next one, and then when it's created, it's also benefiting from its position, attending to each position, which is also becomes part of the local embedding at each position. Not sure if I explained that great, but it's
okay.
You can think of it. It's something which we, like I said, we did was novel to our paper, and we didn't quite realize how novel it was until a bit too late. These days I've seen other works on linear attention, some of them similar, some not. Like Luna, I think, Linformer. Arguably, CLS is actually very, very different. But yeah. Never mind the fact that if I were doing it nowadays, I would definitely be using Mamba, but that the training tasks a little bit because next token is not as useful for this problem.
Okay, interesting. So do you think, I guess you said you will use Mamba more, but do you think like this is specific to like proteins, or do you think it will be useful for like
I have no
text?
doubt that it would work very well in other tasks. Okay, here's the code. I have zero doubt it will work well in other tasks, but it depends on the task. Like say in our situation, we didn't try one specific task of local structure prediction of contact maps of is this position 47 in contact with position XYZ, partly because honestly it was a pain to implement and to evaluate. But also I would assume that in that task it would work a little bit worse. Never mind the fact that for that task you would want to just use AlphaFold, which for structure is the bomb.
Gotcha.
But this is the type which again for some this is very, very good for global tasks, I would argue, and some local tasks. But for some things you want, if your problem is there should be a punctuation or is there a spelling mistake here, that you really want to very, very closely attend all of the words exactly around this one and word number 47. Or say if your task is what is the 17th word in a sequence? And for that, you need very, very specific resolution. If your task is this a well-written document, then you want a very high global level of understanding. If you're taking the average of embedding of all of the words, then a global level of understanding is very good for getting, you will get an embedding of high quality. I mean, I know that I found an embedding specifically for misspelling and another one for a specific type of misspelling, basically the type of people who are not exactly very not very nice people in terms of their misspellings is what I can say. Basically, you can find those type of specific dimensions and learn that in a very good way. So again, it really, really depends on the task. If anything, I would say you could try both. Frankly, it's in my mind, it's very appealing.
Got it. Thank you.
So honestly, I wouldn't want to bet money on it beating the whole, let's attend everything to everything, every single position, and just throw more and more compute at it. But these days I've seen more stuff like the global sliding window attention I've seen from, I think Mistral is quite interesting in this regard. That's a bit more local. There was also a big bird which we wanted to implement but didn't manage to get the whole working with their pre-training and so on. Also shared here, by the way, the code and the Keras, to the repo with all the Keras TensorFlow code. And like I said, there's also a link to the PyTorch stuff if you want to play with it. The global attention thing, I think it has potential, especially if it were done with just a little bit more oomph. Like this was my first work thing with deep learning. Very interesting learning experience. Any more questions? Like the next stuff is diving into anomaly detection. It's a little trickier. Yay, nay. Yay. Okay. So, let's see. So I'll try to go over this a bit quickly since I don't want to tire you guys out. So I'd like to talk a little bit more about some recent works that we've done using protein language models and in the context of scientific discovery and anomaly detection. So the first is a work just accepted on detecting anomalous proteins using deep representations, or basically we want to do anomaly detection. What's an anomaly? It's finding weird cases. And how do we find them? Finding something which is weird means at least we can start looking at them, and there's still a lot of work of why are they weird, what makes them weird, and so on. But it's still easy in saying, well, out of these 100 million proteins, let's find the ones which are problematic or weird or strange or which you want to analyze. So what we try to do is an anomaly detection approach. We're using protein language models to find odd proteins, so odd proteins in various contexts. Because again, it's like, can be odd short? Is it vital? Is it full of static? Is it covered in poisonous toxin? Does it turn everything around it into a hideous zombie disease? Lots of weird things. So in short, what we do is, again, we use protein language models and an anomaly approach based on density estimation, basically kind of KNN, to score proteins on how anomalous they are, either at the local level, i.e. for every single position in a token, in a protein, or the global level, which is just, here's this entire sequence, how anomalous is it? And we do this on lots of different domains and tasks, like finding viruses, prions, enzymes, proteins which interact with a lot of other ones, disordered regions in proteins, and also finding protein domains, and then the proteins of unusual functions. We also, by the way, have a normal full approach there and how we do it, which is actually really useful for other tasks. It does better than meat embedding, which is a pretty high threshold. So one of the things we looked at were prions. Prions, basically mad cow disease, also kuru, aka if you eat someone's, there's a reason humans don't eat other brains. It's not because brains aren't tasty. I like brains. I am a neuroscientist by training, but because we can get prion disease from it, which are nasty, nasty things. Where basically a prion is a protein where basically instead of being like this, it's like this, but it's actually this. And when another protein sees it, it will start folding out. So instead of like this, like that, and next thing you know, the other proteins do it also, and you die horribly, like really horribly. There is no cure for stuff with prions. They are nasty things, and nobody really knows why it really happens. We know that if you have a prion, it causes other proteins to misfold, and they cause others to misfold, but we don't know what causes this chain reaction, which is a pretty crazy chain reaction. It's like saying, it's worse than a zombie virus. We know that funguses and zombies are relatively well known. So what we did was, one of the tasks we had was trying to find prions, where again, there's a tiny amount of prions known. There's like a few single digit to dozens, and most of them are, well, here's the 7 cases, but they're all a tiny variation on the exact same one from cows, and another 5 of the pretty much the same one, plus minus 1% difference from Creutzfeldt-Jakob. It's like really, really few cases which are known. So it's very, very hard to start to detect it, even for few shot or the like. And so again, we did a bunch of examples. We look at their structure. We find a bunch of novel candidates from baker's yeast and the bacteria, which is kind of scary. I didn't really love finding it out. It's like, hey, so there was some proteins in yeast, which if they go a little wrong in your bread, it will cause you to die horribly. And there's nothing we can do about it, because prions are very stable also, by the way. They can persist a long time outside of the body, and they're like, it's kind of like saying, hey, did you know that if a bat with babies scratches you while you sleep, in 10 years you might have babies and then you're going to die? It's like, wait, what? Like, it makes you not want to go to sleep outside anymore. So that was interesting. But the structures again from AlphaFold, very nice tool. And we did the evaluation of various models. I would note the baseline was n-gram features, a bag of words and the like. We compared it to max pooling, mean pooling, and set features. Again, taking the protein, the language model, do the embedding, max pool, mean pool, etc. Set features are a novelty by my co-workers here, where basically take a histogram of the distribution on each embedding vector, on each vector of the embedding, and the max and min. So instead of doing the average per dimension, you'll do, say, 16 buckets, and the max and the min for each one, which lets you capture the distribution a lot better. It's a few more feature vectors, but it beat the mean pooling in pretty much most of the tasks. And that also works for them very well in computer vision. So it's a very nice method, I have to say, like probably like really useful. Like mean pooling is a very, very strong baseline. Like it will beat a lot of stuff. I mentioned, by the way, that we looked at local anomalies, like per position. So we found a really cool thing. So in proteins, we have something called domains, which are basically, they are kind of like functional units. There are various families of domains, and they are the functional units. It's like kind of like saying, like, there is the book, and there is the story of the book. The story of the book would be more like the domain. Like there's a stuff before it and around it, and a book might be a book of short stories, but the domain is the interesting chunk, and we would like to find more of them. Like find me all of the domains for science fiction stories or the like. And we found that the anomaly scores are very good at partitioning the start and the end of various domains, which is interesting because finding a domain is not something which we know in advance, and there are many domains which are unfound. So an unsupervised approach for this is really interesting. I'll kind of skip over it since we're kind of running out of time. I'll say just one last work about looking at viruses. So this is currently under review. So viruses, well, again, I'll leave the 6th time at the end, so I just want to finish the last bit. So viruses, what they kind of would do in order to survive is they adapt, and they basically try to look like our proteins in our body and our immune system, since our immune system wants to kill them, and viruses want to make more viruses. And various viruses which are quite good at this tend to be the ones which we really don't want to get. HIV, AIDS, herpes, or they can also cause cancer or immune system, because once they're there and they stick in there, they just keep on sticking in there. Herpes or the like, it can persist for years. HIV is for life. And so when they're very good at mimicking, that makes them just, again, we would like to understand that better, because it can affect how can we design drugs? How can we detect them? How can we improve the immune response to them? And also, something which is very near and dear to me is the fact how similar is how the biological immune system, proteins and viruses versus the language models. From computer vision systems, how adversarial examples, like this is a giraffe, this is a panda, and it's like we biologically we say, yeah, it's the same picture, but the computer vision is saying, no, this is a stop sign, I need to stop and make the car explode. So like does the same thing happen here?
One quick question. So sort of this section, one of the important things in with viruses is identifying cleavage sites. So like this would be, well, something that would essentially be an unusual position in the protein. Did you ever try to use the previous model to identify cleavage sites?
No, cleavage sites we actually looked for basic cleavage sites for neuropeptides and pro-hormones actually. In viruses, we didn't look at those specifically.
Okay.
The model might have probably would have picked some of those up because there are some families and some traits, but we didn't look at that specifically. So yeah, basically trying to understand what are the viruses which mimic the immune system. So what we did was train models to predict between, this is a supervised approach as opposed to the unsupervised anomaly detection one earlier, to predict between human proteins and virus proteins. So the performance is really high. It's 99.79 AUC, 96% accuracy, which is, it's very high. It's not 100%, and so the question is, how can we understand more about what's going on? So I used the second model, a student model, using an automated machine learning system from my previous employer, Spark Beyond, actually. Let me use a student model using tabular features, i.e. features in a table, and not the deep learning model. We try to predict what are the cases which we had a mistake on. And for that, I used a different approach where I could use lots of features which the normal model couldn't use. The parent model is a whole deep learning model, and that's just only using the sequence. The student model, I could use known annotations. I can use whether it actually is a human or virus. I can use what type of virus family it's from. I can use functional annotations and keywords. I can use the sequence length. I can use lots and lots of features and only the interpretable features. And then my target is, did the model make a mistake or not? I'll also note, just to the TLDR, language models make the same kind of mistakes as their biological immune system, which, but it does kind of suck because it tends to make them about with the viruses, which you usually know in the sense of that you really, really don't want your doctor mentioning those words. So again, evaluation, high results. Like again, just doing the length or just doing the amino acid engram gets decent results, but all of the deep learning stuff just thrashes it easily. We don't compare it to doing a BLAST baseline though, because that's annoying to infer. ESN2 is a model from Facebook, up to 650 million. The linear ones are using the pre-trained embeddings from the 3 billion model I mentioned. Like recently they were made available for a bunch of proteins. It's not useful for doing inference, but for only doing training on the subset of, they did it for about 100,000 protein, or 100,000 proteins or so available, again out of 100 million, but for only training on those, it saves a bunch of time because I don't have the resources to do embedding from a 3 billion model. It's tricky. Also, by the way, the training here I used LoRa, PEFT, Hugging Face. I also tried the recently released DORA for those who have seen it. It actually worked way worse. Don't know why. Also, lots and lots and lots of bugs. There's a lot of bugs when you're using LoRa and PEFT with the protein language models, which aren't, not using a, more popular model. Like it's very buyer beware. Like horrible bugs. Like hey, the saved model doesn't actually save the stuff that you saved, even though it says it does. Type of bugs like loading and reloading it, and it doesn't actually save it or load it, or says it does, but doesn't give us stuff like So anyway, so we did a bit of also, here we see some clustering about, again, humans and viruses, and also the cases where we have the mistakes. This is, by the way, supervised clustering by the protein embedding, so it's even using the information and the trained model, it kind of separates them. They are quite different, but they still overlap, and the mistakes are relatively evenly distributed. We're not seeing a cluster of, oh, all of the mistakes are just here or just there. They're like all over. They're like, different mistakes in humans and in viruses, and then spread all around. There's a prettier figure in the paper, but never mind that. So again, the error analysis model, like I said, is just using a bunch of the extra features to predict we're trying to predict the mistake. This is an explanatory approach because, you know, good luck trying to look at the example from, well, here's the embeddings and here's the 4 closest examples, and that's KRRKP, PKJKRPF. You can't learn anything from that, frankly. In most cases, depends where and what. So basically, we took a bunch of features out of there about what characterizes the mistakes, and you know how many percentage of cases mistakes have them, how many cases they catch, and how much the lift is how many more mistakes do we see in cases characterized by this feature compared to the overall baseline. So if you know 4% mistakes and the lift is tenfold, then well, you know, 40% of cases with this feature have a mistake. And stuff like the adaptive immune, which is basically virus proteins which shift the immune system. Their functions are to find the immune system T cells and just, you know, kill them or fill them with glue. You know, oncogenes. Well, unfortunately, viruses which are known for causing cancer are also harder to detect. Viruses have a lot more mistakes relatively. They are trickier. Endogenous retroviruses is something which I learned about here. It's basically they are a nice argument of false positives or false negatives. They are viruses in the human genome, which are actually virus proteins which, you know, thousands or millions of years ago just got in there and just got latched in, and then the whole genome just needs them from being expressed. So technically they were labeled as humans, but the model said, well, no, these look like viruses to me because they are freaking viruses. So a bit of an argument about whether to keep them or not. We also find differences at the so we use viruses from everything with a vertebrate host, basically everything with a backbone, which includes a bunch of animals and mammals and also humans. Viruses which are specifically related to those with humans as their specific host are more likely to be mischaracterized, and those which specifically target humans, like, you know, HIV and AIDS, humans, it can be humans and monkeys and whatnot, but are specifically targeted at humans, are more likely to be misclassified because evolution's a bitch. You know, the ones which evolved to target us are good at fooling us and preying on us. Of course, there's a bunch of different virus families. Not going to go into this given the of the audience. And also something else for explainability is the fact that in terms of how much they activate the immune system, which again, getting some of the various outputs. So we found that the mistakes from humans and in viruses, virus mistaken for human and human mistaken for virus, are really similar. That regard, they trigger the immune system a lot less. They're not as untriggering as normal human proteins, but they are like much less triggering the normal virus proteins. So they're similar to each other. They're both weird. They're like they share a lot of commonalities, which is interesting. So now we actually have each of the talk, and yeah, questions. As by the way, anyone can see some of the papers, and of course all of the code and whatnot. But you know, yeah.
Thanks for the presentation. I guess like one question I had for the last paper was like, I don't know, like if you present these proteins to like a human doctor, is like the classification rate like 100%?
No, and a doctor won't have any clue what to do with them. A professor of biology, like actually my mentor, she did look at a bunch of them and just could tell some of them apart, though not 100%. Like think about how many you need to get to show 100% versus 96. You need a lot more than 100, and each one is a lot of time. It's like here's on the site called Uniport. You can look at them. It'll takes time, but like some of them, my mentor doctor said this one is. Viruses, some of them you can look at them and say this is weird. Like some of them, even the naked eye, you look at it's like. Okay, think of this way. You know how there's the examples I like to give in Bayes and entropy about heads and tails flip, like heads, tails, tails, heads. If you see like 50 H's in a row, you don't know much, but you know it's like that's weird. That doesn't look right. You know, you can look at it and say that doesn't look right.
Interesting. Good.
So like something, but in most cases it's very, very hard to do. Like not something which is just. Because again, it's 20 letters. It's not like looking at words where you can easily kind of tell them apart. You can tell by the composition to a degree. You can tell sometimes by the length or properties, like really weird properties. But in many cases you can't. And it's, you know, ones that you can. Again, some of them are relatively easy. Some of them are very hard. And even then, it's like, you know, it's not something. If I were to, doctors wouldn't know. Like someone who's in molecular biology could see about some, but if it's like saying, you know, you show it to somebody, they could tell if there is a bug in it. Like some cases, yes, but it's not the average programmer off the street.
Interesting. Got it. Thanks.
How hard to classify was COVID? Did you feed COVID into it? Like
Actually, COVID it got it correct. It's relatively it's not hard. A second, it's Anthony. So actually, I have a different paper currently in review about how I show that COVID is actually should have multiple the whole long COVID thing, but COVID is classified correctly. SARS and COVID it's not hard in that regard. It's the problem there is not the immune system missing it. It's the fact that it's it goes around.
Right. High contagion, not whatever, high mortality.
Yeah, like if we had COVID and it was, or with HIV and cancer, I mean, I would rather just watch a zombie movie instead. It's less scary. Like we got really lucky sometimes, honestly, because there is nothing technically stopping the whole. Hey, why don't we have? By the way, why don't we actually have? Why doesn't your HIV just travel okay in mosquitoes or something like that? Like, because we're lucky and because we haven't had anyone stupid enough to try to prevent we just really case of we just didn't get unlucky. Nothing stopping it from that regard. Isn't biology wonderful?
I was also curious. One of the things when people were first trying to analyze COVID, one thing that confused people is that it had this programmed frame shift.
The furin cleavage site is just kind of weird. Yeah, I heard about that. I never went into the details. I don't know enough to talk about it. I think it's something about the furin cleavage site, which is a relatively common pattern. There's something about where it is. I don't know, but again, it's a case where it's.
It wasn't that there was a cleavage site. It was that the cleavage site is like next to a place where it purposely shifted the codon window. So like you go in sequences of 3 RNA bases. The frame for the ribosome like purposely shifted right before the cleavage site.
So that I don't know about. That you would need to look at a DNA level model. So honestly, but again, there it's not my my forte. Also note that in general viruses they just make lots of copies and you're only seeing the successful ones. So if there's a lot of viruses and pretty much anything else there are a lot of the buggers.
Thank you for the talk.
Pleasure.
Yeah, thanks. This was an awesome talk. If you have anybody else, if anybody has any open source work they're working on.
Do you study ART genes and how retroviruses have written onto those?
ART genes? I'm afraid I don't know what those are. I know retroviruses. I don't know what ART genes are.
The activity regulated cytoskeleton associated gene.
If you PM me about it, it sounds interesting. I'm not a cytoskeleton person. I'm more of a secreted proteins person.
Okay.
Or healthcare. Any other questions?
Here's one final question from me. Is there any way to 100% classify a protein that is known?
What do you mean classify?
Or see if it's a virus or a fine protein. Does it have to do years of research and then you finally add it to the database?
Kind of. It really depends. Like what you would do in general, there's various online tools and bioinformatics pipelines. What the rule of thumb would be, just run it through a pipeline, like say, ask what is the most similar sequence. If you have here is a match which is 99% similar, protein XYZ, which does A and B, which is well known, then you can kind of say, yeah, this is what this protein does. If it's well, here is a match with 1E3, then that's which is really terrible. And it's saying, well, then you can look at domains, then you can look at other things, and you can try computational annotating pipelines, which some things they work well, for some things less well. It's really depends on the what. some cases you might be able to annotate it very well and very automatically. In other cases, it might be, well, we have a protein, we don't know what it does, and it might be a novel function, in which case it's like, well, that sounds like a good thing to do your PhD on for the next 7 years or so.
Gotcha.
It depends, but you know, if you have something like totally not like, you know, CRISPR and Cas9 is an example for this. Like, actually, no, it was a known function. Like saying, you know, that clips DNA. Here's another one, and we think it works a little differently. So it's that clips DNA, and hey, we can manipulate it, and then next thing you know, you have a CRISPR and Cas9, which are like, you know.
Gotcha. Yeah.
I was just curious to know, like, how old are retroviruses since they were introduced early on with the eukaryotes? Is there any way to know?
I don't remember. Like, if you told me to guess, I want to say let's probably millions. I mean, hundreds of thousands is way too early, but I can honestly, I would open the Wikipedia for that.
Okay.
I mean, there's definitely an extinct from give or take bunch of them. Yeah, I don't know. So basically, there's at least from the level of vertebrates. So that's like millions, billion. Like been around for a while.
Right.
Anyone has any more questions? Okay. Thanks a bunch for the presentation. I think Adam's gone, so yeah. For next presentation, I don't think it's decided, but hope to see you all there. But yeah, again, thanks a bunch for the presentation.
Thank you for having me.
See you guys.